first part will be focused on RdRp inhibitors. RdRp represents RNA-dependent RNA polymerase. Here we can see the sequence of the RNA polymerase and the structure, protein structure. RdRp is also known as NSP-TRF. It can catalyze the synthesis of viral RNA. It has been viewed as primary target for developing antivirus. Normally, those drugs are nucleotide analogs. Here we can see some representative drugs has been used to fight against novel coronavirus. We can see 4. The first, remdesivir. The second, favipiravir. Third, ribavirin. Fourth, panciclovir. Those different chemicals has been approved for different indications, but they share the similar mechanism. All those chemicals works as prodrugs. That means it will be transformed into active forms in vivo. On the lower part, we can see normally it will be transformed into nucleoside triphosphate. And then this triphosphate will be taken by virus. Because it's an analog, not the true building block for RNA synthesis. In turn, it will block the RNA synthesis of virus. Here we can see a general mechanism of action of how RdRp inhibitor works. This red arrow shows the RdRp inhibitor will inhibit RNA polymerase involved in viral replication. As a result, it will stop the synthesis and the virus propagation. The first nucleotide I will discuss here, the name is remdesivir. It's developed by a company called Gilead. The chemical structure is shown on upper right. It's an adenine analog. Originally, it was developed for treating MERS and SARS. It has been tested in vitro assay, has an EC50 value around 0.8 micromolar. Here we can see the mechanism, how it works. The first, remdesivir will be hydrolyzed through the breaking the ester bond to get this alanine metabolite. Then the alanine metabolite will be converted into nucleoside monophosphate. This monophosphate will be further transformed into triphosphate. Finally, this triphosphate will be the active form to stop the viral RNA polymerase activity. This drug has been proved to be highly active against Ebola virus in rhesus monkeys. Here you see the data. This cartoon shows the incorporation model between the RNA polymerase of novel coronavirus and the drug remdesivir. As you can see, here is the diphosphate structure. They have the hydrogen bonding interaction with the amino acid residue from the novel coronavirus polymerase. And this drug has been tested in a clinical trial. On the top right, the remdesivir can improve the overall outcome of the patients. On the lower right, for patients either non-invasive or invasive, the drug can give the improvements. Second nucleotide analogs I will mention here is favipiravir. Here is the favipiravir structure. It's a guanine analog. It has been approved for treatment of influenza infection. The in vitro assay shows this drug has an EC50 value around 60 micromolar. Here, you can see how it works. The drug will undergo phosphoribosylation to get this RNP intermediate. Then this intermediate will undergo further phosphorylation to form this active form. And then this active form will block the RNA polymerase activity to stop the viral replication. In the late of last month, the company of Fujifilm has announced to start a phase 3 clinical trial by using this drug to treat COVID-19. As you can see in this table, this drug did give some improvement for patients. The third drug I will discuss here is named ribavirin. It's a rather famous antivirus. Here's the structure. Ribavirin is also a guanine analog. Actually, it's a broad-spectrum antiviral. It has been approved for HCV and RSV treatment. It has a moderate activity, which is around 100 micromolar for EC50 value. The true mechanism is rather complicated. Other than the major contribution by inhibition of RNA replication, it was proposed that ribavirin can work by other pathway. For example, like it can inhibit IMPDH. In addition, it also can work on RNA mutagenesis. At last, it was believed it can work as a modulator for immune system. So by those possible mechanisms, ribavirin has been used in the clinical trial for treating COVID-19. Here we see some listed clinical trial using ribavirin. The first is the ribavirin with other drugs. It's conducted by University of Hong Kong. And the rest was clinical trial conducted in China by either using sole ribavirin or the combo with other drugs. The fourth RNA polymerase inhibitor I will mention is panciclovir. Here is the structure. It's an guanine analog. This drug originally is approved for herpes virus treatment. The EC50 value is around 100 micromolar. The mechanism is similar to the other three. It will be converted into the triphosphate form in vivo and then block the viral replication. Not surprisingly, due to the efficacy of nucleoside analogs as potent antivirus, people have started with deep learning-based computational discovery to find other nucleotide analogs. On this slide, you can see quite a few nucleotide analogs. Because of time limitation, I will not go further with all of them. But I believe some of them should be also effective for potential treatment.